Did you know that a simple act such as waving your hand or moving your arms consists of an interesting yet complex system within your body? You may not realize it, but your body works hard to do these simple actions we take for granted. Today we are going to explore the system in the body known as skeletal muscle. This is what makes all your voluntary movement possible, and when we are done, you'll have a better understanding of how a simple movement is more complex than you think. Let's begin, shall we? As you can see in the model, the muscle is contracted. If we zoom in closer, we can see the whole muscle. Within the muscle, we see the fascicle, which is a bundle of muscle fibers. The muscle fibers are surrounded by mitochondria, which makes it possible for ATP. If we zoom in closer and pull one of the fibers out, we see it is covered in cell membrane known as sarcolemma. The sarcolemma is made up of transverse tubules, or T-tubules. The sarcolemma and the T-tubules are what receive the action potential to begin muscle contraction.
Action potential is the electrical current that goes to the synaptic knob. Once the action potential arrives in the synaptic knob, it stimulates the synaptic vesicles, which hold the neurotransmitters known as acetylcholine. The vesicles release the acetylcholine, which once released, the acetylcholine crosses the synapse, which is full of sodium ions, and into the acetylcholine receptors that are on top of the muscle fibers. Once the receptors are full of the acetylcholine, creates an opening for the sodium ion. Once the receptors are full, the acetylcholine creates an opening for the sodium ion channels, creating an action potential within the muscle fiber.
Though once the acetylcholine is released and fills the receptors, it needs to be able to be removed from the receptors. If not, the muscle will continue to contract. In the synapse, there is an enzyme called acetylcholinesterase. This breaks down acetylcholine.
Now back to the action potential. Once it reaches the muscle fiber, the sodium ion channels open. This allows the sodium channels to enter the muscle fiber, entering into the sarcolemma. This then allows the action potential to occur in the sarcolemma.
Once this happens, the action potential then travels down the sarcolemma down to the T-tubules. This stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum. Calcium is sequestered by calsequestrin, where it is stored in the terminal cisternae.
Now before we go any further, there is a structure we must go over in the muscle fiber called the myofibril. This myofibril contains thick and thin filaments. These are what make up the structure known as the sarcomere.
Sarcomere is made up of two proteins, actin and myosin. The ends of sarcomere are the Z-band, that is the attachment site of actin. The I-band is a ring of the sarcomere that contains only actin. The A-band is where the myosin attach. Last, the H-band is a part of the sarcomere that contains only myosin.
Now that we have gone over the structure of the sarcomere, we can continue back to where we left off with the calcium. It is with the sarcomere of the myofibril that the calcium floods out and binds to the thin filaments known as the troponin. The troponin is pulling off the myosin off the active site of the actin. The active site exposed leaves the open for myosin head to bind and create the cross bridge. The myosin then pivots to lower energy. Troponin is pulling off the myosin off the active site of the actin. The active site exposed leaves it open for the myosin head to bind, creating the cross bridge.
The mitochondria releases ATP that then binds to the myosin head. This then causes the head to break from the active site on the actin. Once this happens, the myosin head is energized and the ATP is converted to ADP. Then later, the calcium is pumped back into the sarcoplasmic reticulum. There it is put back into the calsequestrin, where it is stored until activated by the flow of calcium again. This allows the tropomyosin to cover the active site and then muscle can relax.
So like we said before, simple movements we take for granted are all part of a complex yet unique microscopic system. We hope you enjoyed and now have a better understanding of muscle contraction.